Hello, and I guess this is the second part of the week 6 lecture for the next generation sequencing class. This is a relatively short um, set of material that I just want to go over, in part to help you with your homework, including getting familiar with the VCF format and kind of providing you with a small introduction to the GATK or the Genome Analysis Toolkit website. So I left off, however, I think in the last lecture, mentioning how we go about annotating our SNPs. And what I mean by annotation in this context is that we are taking the annotation available for a genome that gives what we call the structural annotation about the positions of various features in the genome, such as introns and exons, and we're taking that, which typically, as you know, comes in, say, a GFF or a GTF format, and we are then applying that to our SNP data set so that we can assign sort of basic functions to our SNPs, such as what class of mutations they belong in. Are they non-synonymous, meaning that they alter the amino acid? Are they synonymous, meaning that they're at the third positions of codons, for example, but do not modify the amino acid, whether the SNP is in an intron, etc. And so SNPF is sort of the method of choice for doing this. We do this, in the 1000 Genomes Project used SNPF, in their project, and it's sort of carried on now, so that I think it's fairly common for different research groups to use this program. So this is a separate program under development. It's been co-opted by the GATK now, as they've integrated into their workflows. So a SNPF is essentially just it's a software package that builds a database of the structural annotation from a GFF or GTF input. It then allows you to apply that database to your SNP data set, and in the end, what SNPF does is it gives you the annotation for individual SNPs in a VCF formatted file. So these are just a few of the predictions that some of them I've mentioned, that SNPF will make. And so this allows you to kind of quickly sort through your SNPs and sort of rank them in terms of candidates of ones that might be more damaging to a cell compared to others. So anyway, I'm hoping to have a chance to have you guys incorporate this somewhere along the way, either in your the SNPs you're working with next week, as you've called SNPs now in your homework this week for week 6, and I'm thinking about how I want you to then handle those SNPs in your next week, week 7. One option would be to work with SNPF. So we'll see if I can't come up with a way for you to easily sort of integrate this into your analysis. Now, I did want to go back to the variant call format. This is the format. I did mention this going all the way back to week 1, but at the time, I'm sure I really wanted to expose you to some of the common formats and to kind of show you, express to you how essential the formats are, how rich some of them are now. But here, now that you're working with them, I thought I'd take just a moment here to go back, since you'll be working with VCF in your week 6 homework. So remember, the VCF has a header. It has what is called the body. Header always begins with these hash marks, and notice this one special header line here, which is actually shows which information is in each of the fields in the file. So position 1 or column 1 has the chromosome, etc. Here I'll point out that that VCF is meant to be a single or a multi-sample formatted file. So in this case, I'm showing you 2 sample genomes that have been sequenced in this experiment, and each sample genome will have an additional column. So if you had 100 sample genomes, each of these samples would appear in 100 columns after the initial 9 columns that are sort of the fixed dedicated columns in VCF format. I also want you to remember that for the info, the format, info and format fields, that there's also tags here that indicate information about those fields. It looks like alt here also has information in the header. So this is the alt allele column, although I'm not sure that this is always this is a standard, or at least that it's it may be flexible and allows it. I'm not sure it's absolutely required. In any event, you can see descriptors of the info and format tags. Okay, so GT and DP here mean that in the individual sample columns, the first subfield GT is here. So this is before the colon in the sample field. GT is the information that's in the first subfield of each sample. So we can go back up here and look, okay, what is GT? And GT here means genotype. That's the description given to it. In this VCF, DP here means, if we go up here, DP in the format field means read depth. So this is a sample-wise read depth here now, and we can see that read depth in sample 1 is 13. Of course, I'll just mention that this is sample-specific information, so these are what we call the genotypes, right? Remember I described in the part 1 of this lecture that we have genotypes and we have SNPs. So there's genotype calling and there's SNP calling. The row in the VCF is the SNP, or the variant, whereas in the individual samples we actually get the genotype. Okay, so I'll also mention that we encounter both what we call variant-only VCFs. These are VCF files that only have variable sites in them. We also can store all of the information, in the from a data set, including invariant sites. So you can store the entire genome here from a sequencing project. However, I will point out again that one of the main reasons to do resequencing is to get the variable sites, and people are less often interested in the invariant sites. And so for a variety of reasons, people just tend to work with the variant sites, although as you'll see in the GATK, when you do the SNP calling, you can also call invariant sites. So you do have that option. The file obviously gets very large when you include both variant and invariant sites. So this is a variant-only VCF. Now, I'll also point out that there are different types, not just SNPs. Different types of variation can be stored in a VCF. So here's one way of indicating a large structural variant, and this is a large deletion. And as you'll encounter in your homework, I show you how small insertions and deletions are handled. So this, I'll leave it to you. I've left in the homework a link to the VCF specification. I encourage you to read that. But one other thing to kind of just get you going here is that this is the reference allele here, this is the alternate allele here. The point is that if we number these alleles, okay, this is the 0 allele always, this is allele 1, and this is allele 2. This is allele 0 and here in the second record here, this is allele 0, 1, and 2. This is the most standard situation where we just have 0 and 1. So let's go over here and look at 0 and 1. Here we have in the genotypes, we have 1 and 0 and 1 and 1. Okay, so the point is that this 1 and 0 then reflect, you can actually extrapolate what the actual allele is, based upon the ordering in which they appear in the ref and alt columns. Okay, so I think most of what I wanted to say about it. I think the rest is fairly straightforward. And so I guess the final thing I'll say, I did forget that info column basically contains information about the variant, okay, or the entire record. It oftentimes includes the depth. It has, you know, it's a very flexible. VCF is flexible, so the info column doesn't have requirements per se. So you'll see people include different types of information, but you'll see as you go through the homework, the different types of information by default that the GATK anyways in its snip calling dumps in the info column. Okay, so that's kind of all I wanted to say about that. What I'm going to do now is jump over to the GATK website. So this is broadinstitute.org/GATK. And their website is really, a place where I spend a significant amount of my time, because it's a place where there's active development, there's new tools, there's things that, simplify my job in working with some of these large resequencing data sets. And I just want to point out, if you go to the main page, you can, if you click on Guide, this is where you normally will go. I find these here, sections here on the left to be particularly useful. So one thing, as you're starting out, they have a nice section on intro materials. Many of these are getting GATK to run effectively on when you're needing to use parallelism or working on a cluster type system where we have shared resources and getting it all configured. So a lot of this is not important for your purposes, because you can easily just download it and execute simple tasks or smaller tasks without needing to work on a larger cluster. So I invite you to peruse that section on introductory materials. There's also a nice section on methods and workflows. So, for example, best practices for variant detection with GATK. You'll notice that they oftentimes have archival sort of copies, because this is actively developed software, and things change, and so they kind of update you with what they consider to be best practices. I'll also point out that GATK is heavily human genetics oriented. So this is really came out of the 1,000 Genomes project originally, together with Picard tools, which is also developed by people at the Broad. And so this is heavily geared towards calling variants in human data. And as I pointed out in the lecture, sometimes there's a lot of issues we encounter when we're not working with human data. So some of what I presented for how to call SNPs is a little bit simplified, simply because getting everything going with the variant quality score recalibration and the base quality score recalibration is kind of beyond the scope of this class, but something that you can implement, and there are tools to implement right here at using the GATK. You might also check out the technical documentation. So this is where you find sort of a lot of tools. Basically, these are command line programs then that do various things. So let's take one simple example. This command line program just selects the header information from a VCF. This one here is one that I'll have you use. It's called variant filtration. This is how you actually would apply filters to the filter column in the VCF, which I neglected to mention, but we'll be working with. This is the basic strategy is to apply something, a tool like variant filtration here, which then, here's the standard example command line for variant filtration, and here they give you details about the individual options. But this is the way that you apply filters to a VCF where you're marking individual records or rows, that is SNPs, that meet certain criteria, like the overall depth was a certain level or below a certain level, the variant quality was not up to standards. You can mark that and indicate that, and then you can use another tool called select variants, which allows you to then actually conduct the hard filtering if you desire to remove certain classes of SNPs from a VCF. So I'll be showing you how to do that. Here's another nice one, variance to table. That just gets, basically dumps all of the information from a VCF into a table. And that is a useful thing sometimes. So oftentimes I'll just want to dump something quickly into R, which would be very difficult to process, or there isn't a tool available to process in the VCF. So I'll dump it to a table. It's just a tab delimited table. I can read it into R. I can plot or extract information, which you already have a sense of how to do. Anyway, that's just kind of an overview of the things I'd like you to do, to have a look at. I didn't show you the best practices, of course, reading that. I'm trying to, in this course, give you what, I consider to be best practices, but the GATK for human data has their own set of criteria that they think are very useful, and I would default to them because these guys, are among the best in our field, and if it's good enough for the GATK kind of thing, it's good enough for us too. So. In any event, that's kind of a tour of the GATK. We'll be talking more about it both in your homework and going forward, where I'll have you applying some of these methods to familiarize you with them. So with that, I think that's all I wanted to say for the week 6 lectures. So good luck with your homework, and we'll see you next week.